I would like to welcome you to our final insights for the spring season. It's an interesting and thought-provoking lecture that we will be receiving this evening. We have Professor Michael Pollastri from the Department of Chemistry and Chemical Biology, and he's going to speak to us today on Going Green, Green Chemistry and Neglected Disease Discovery, Making Molecules with a Conscience. Tonight, we are also honored to have the founding dean of the College of Science, Dr. J. Murray Gibson, this evening. Prior to joining Northeastern, Dr. J. Murray Gibson was Director of Advanced Photon Source at Argonne National Laboratory in Illinois, where he served as Associate Laboratory Director for Photon Sciences. At Argonne, Dr. Gibson spearheaded and secured funding for two major initiatives: the Center of Nanoscale Materials and the Transmission Electron Aberration Corrected Microscope Collaborative Projects. Dr. Gibson earned his Bachelor's of Science in Natural Philosophy at the University of Aberdeen in Scotland and his PhD from the University of Cambridge in England. Please join me in welcoming Dr. J. Murray Gibson.
So I go by the name Murray, by the way. It's nice. Thank you for coming tonight. It's a pleasure, actually an honor to be the founding dean of the College of Science. I think some of you who graduated from departments in the College of Science did so when it was called the College of Arts and Sciences. Can you hear me okay without the microphone?
Yes.
Yeah, okay, I think so. When it was the College of Arts and Sciences, and the university made a decision to which I think was a smart one, to take that college and the College of Criminal Justice and make three colleges. Arts and Sciences, the College of Arts, Media and Design, and the College of Social Science and Humanities. I think what's really was important about that was the university is in a fantastic place right now. We got more applications than any other private school in the United States this year, 43,000 applications for our freshman places. And the quality of students, incoming students, is really outstanding. And because of that, because of the demand for places here, and we are in a position to recruit people. We have a number of faculty who are retiring this year. This year alone, I think in the College of Science, there were 6 or 7 faculty retired that we're replacing. And also the university's made a commitment to grow by about 10%. So between those two, there's an opportunity to hire quite a number of faculty in the next 3 to 5 years. And so I think that alone is an important reason to have deans who are closer to the ground, who can work with the faculty to figure out whether, science is a dynamic field. It changes constantly. You have to really be, working closely and thinking about where science will be in 20 years from today. Some of our very strong programs today, like behavioral neuroscience, didn't exist 20 years ago. And so we just have to be very, very nimble. I think we have a great opportunity to position Northeastern in a very great place in the future. Why is Northeastern so successful? I think it's, in my mind, I came here only 6 months ago. And what attracted me was the experiential education. This is unique. Northeastern's strong history and co-op, combined with now a very top-notch research university, puts us in a unique position to offer the kind of education, which particularly in science is so critical. That is combining academia with the real world, helping people to figure out what they're good at, to end up with a career that they're where they have a passion and to be successful. So this is a great place to be, and we're very engaged in strategic planning at the moment, thinking about the future of the college and working with the faculty on ideas for areas where we can excel. I mean, my attitude is what we do should be the best. And one of the great things about being in Boston is there's an incredible opportunity to partner with industry, partner with other academic institutions. So we don't have to do everything ourselves. And I think our speaker tonight, Michael Pollastri, in fact, had worked a large part of his career in industry, working for Pfizer. And we have very, very strong connections between pharmaceutical companies, biotech companies, and I think it's something we can highly leverage as we move forward. It enables us to stay at the cutting edge. I grew up and went to, after graduating from the University of Cambridge, I came to the United States 35 years ago, and I'm a citizen. I came here because nobody else was giving me a job at the age of 25 where I could actually do whatever I wanted. And I'm serious. That's the great thing. The US is really great at entrepreneurship. supporting young people, giving them the ability to be successful. So I got a job at Bell Laboratories where I was in a position to go off and do whatever I thought was most exciting. And after 3 years, they checked, and if I'd done okay, they gave me more money. You wouldn't find any other place in the world that had that sort of environment. But it's really a key thing that I think I wanted to mention, that that environment was a highly disciplinary lab. And I learned something there, which I think is critical here too, that you often benefit. A lot of academics think, well, you know, ideas come in academia, they flow out to industry and make products. But what I learned in my role there, where I was doing pretty fundamental research related to semiconductor science, semiconductor physics, was it works the other way just as much. Sometimes industry has tools and ideas that are way beyond the imagination of academics. And then from that comes the other direction, which is ideas to do things academically, creatively, fundamentally, that you never could have thought of or would have done. So having a very strong connection between the real world and the academia is something that Northeastern is very well poised to exploit, and that's one of the things that's attracted me to come here. So I'm very excited, and I think you're going to hear one example of one of the strategies that we see as a tremendous opportunity for the future that Michael talked about. He promised me there wouldn't be anything gross in the talk, right? So you can eat your food and not worry about seeing things. But I will say that drug discovery is something that the university is already well known for. We have centers, a Center for Drug Discovery with Alex Macrianis. We have Lana Tortula, who's very well known around the world for his medicinal chemistry, nanotechnology, drug delivery. We have others within college of science. Mike has come in here and has taken an approach which we think is very exciting for the future, and that is he's focusing on what I call diseases that kill people with no money. And it turns out, if you look at it, and Michael probably showed you statistics, a whole lot more people die of those diseases than die of prostate cancer, for example. But sadly, we put trivial amounts of money into solving the problem. But what Mike will tell you is there's approaches he can take based on his experience in the pharmaceutical industry, which can repurpose drugs that were developed for other purposes to try and address this massive problem facing the human race, which are many, many, I guess the word is orphan diseases. So I'd like to introduce Mike, and he'll tell you about how this can be done with green chemistry.
Thank you. It's really an honor to be here to speak to alumni. I've only been here at Northeastern for just shy of 2 years, but I get the sense that the alumni network is very strong, and so this is a good example of that, I think. Just a little interaction while you're still eating. How many of you were science-type majors at Northeastern? How many of you are scientists? Okay, good. How many of you are currently still practicing science? Okay, and how many of you have taken chemistry at some point in your life? Professor Engen has taken chemistry at one point in his life. I'm glad to hear that. Okay, so the first word I say is chemical. What's the word you think of?
Good.
Oh, no. You're not a good setup. What's the public, the general public? You say chemical, it's usually preceded by toxic, right? Because chemistry has an image problem. When people talk about chemistry or you see stuff on the news, they frequently talk about a hazardous chemical release or a toxic chemical. And so we have issues such as this happened about 25, 30 years ago in Bhopal, India. 25,000 people were killed because of a release of methyl isocyanate. So here's a poor part of India. You have a chemical plant right behind it. And this huge cloud of toxic gas came over and killed 25,000 people, and they still have the effects of this today. Anyone recognize this picture? This is up in upstate New York. This is a town called Love Canal, right? This is Love Canal. So if you're actually watching this on two screens, I'm using the laser pointer on this one closest to me, just so you know. So Love Canal was an area where toxic waste was dumped by a chemical company over the period of many, many years. And then they capped it. They capped the landfill and then sold it to the city for $1, the school department for $1. They then built, over the period of the next couple of years, schools and houses and a neighborhood. And then over the next couple, say, 10 or 15 years, people would be digging and they'd disturb the top of the cap and release chemicals out into this hazardous waste would come up. Heavy rains for one season brought all these chemicals up into people's basements. People started getting cancer, started people getting sick. And it ended up being, actually, in the late 70s, the beginning of the Superfund program, which you may have heard of, put into existence by the Carter administration to clean up toxic waste as it shows up. And this is a sort of, the kind of horrible train accident with these tank cars carrying chemicals. You see this right next to the river. Holy cow, what a horrible thing, chemicals. And more recently, you look at these bottles. I'm a parent. I have 2 small kids. We use these bottles all the time. Both of my kids use these bottles. These are polycarbonate plastic. It's hard and it's clear. But now, as a process of the production of it, they have contaminated with a byproduct of the polymer formation called bisphenol A, which is a endocrine disruptor. So it has these other effects. So this is we can now have to buy BPA-free bottles. So these are the kinds of things that we frequently hear about in the news. But when you think about it, plastic furniture, your plates, makeup, pigments, drugs, it's all chemistry, right? These are all chemicals. These are all things we use every day, and we sort of take it for granted. And the image problem is this, because sometimes you have chemicals like this, which are pesticides, in use. They give us a food supply that is safe and copious. If we didn't use pesticides, we would not have the crops that we have. This is a truck spraying DDT back in the 60s, right? And these are kids, by the way, chasing after it because they enjoyed the smell. DDT eradicated malaria almost entirely, okay? However, used to excess, it created resistant parasites and caused what eventually ended up being the beginning of the environmental movement. It killed birds. Their shells got really thin when the birds were exposed to DDT. And then this is a picture of a granola bar label. In green, you have all these chemicals. These are all chemicals, right? So folic acid, riboflavin, thiamine, vitamin B1, and then natural and artificial flavors. Chemicals. They're not bad, right? People demand it. They like their stuff they like their red fruit filling to be red and a little bit purple, so they put a little blue in it, right? So really, everything's a poison, depending on the dose, right? So if you take anything to excess, it can be a poison, right? And so this is the problem that chemistry has, is how do we make the general public aware of the fact that there's a lot of good to come out of chemistry and chemicals. We use them every single day, but we don't do a good job educating the public. So as I mentioned, the environmental movement was launched in 1962 in response to the widespread use of DDT. Rachel Carson published this book called Silent Spring. This is the cover of the 40th anniversary edition, so it's been around for quite some time. So this was published in the early 60s. And basically, the Silent Spring refers to the sound of no birds. That's what the Silent Spring was. And that launched the environmental movement. And the result of this was legislation. So if you start in 1870, so what we're plotting here is the number of new laws in the dark line, and then, the new laws are in the red line. So new laws come up every year or so. You see a lot of activity after the 60s, after this book came out. And then you see this ramp up of cumulative environmental laws. So this is basically the government regulating how chemicals are handled, how chemicals are disposed of, right? It's all good. Don't get me wrong. This is all very good. However, chemistry has to come back to this and say, well, how do we respond to this? How do we take advantage of the benefits of chemistry while we minimize safety concerns to the people who work with them, like me, the and the general public? How do we maximize the benefit and minimize the cost? And so in the late 90s, John Warner, who used to be at UMass Boston, and Paul Anastas, who is currently at Yale University, developed this concept called green chemistry. Green chemistry, they literally wrote the book. The practice of utilizing a set of principles to reduce or eliminate the use or generation of hazardous substances when we design, manufacture, and apply chemical products. Okay? And what this did was it laid out a set of guidelines for practicing chemists and product development folks for how we can start to do chemistry in a more environmentally sustainable way. So they came up with what they refer to as the 12 principles of green chemistry. I'm not going to go through a lot of these in detail, but I'll give you some ideas. One is prevention of waste. Don't generate waste unless you have to. Obvious, right? Less hazardous chemical syntheses. This is directed towards people who work in labs or work on production plants. Things like design for degradation. So we buy a product, we manufacture a product, we sell it. Someone buys it, they use it, they throw it away. What happens then? Does it just sit in a landfill? Does it degrade? If it degrades, what does it degrade to exactly? Is it something worse? So you have these 12 principles of green chemistry, and you can bin them into really two areas. One's environmental impact. So in terms of waste prevention, where do we get the stuff from? Is it renewable feedstocks, or is it from petrochemicals? Waste generation catalysis, how can we do more efficient chemical reactions, and what happens to the stuff when we're done? And then worker safety in the purple boxes, the people who use the chemicals in production, what do we need to think about? How do we do stuff more safely? How do we analyze for pollution prevention? So we have a big, huge production plant. How do we make sure that we're not emitting something into the environment? And then inherently safer chemistry. So you can come up with the nastiest chemical process you can imagine, but there are probably safer ways to do it. So why do it in the riskier way? Okay? So here's a chemistry primer for those who haven't seen chemistry in a while. We take two reactants, say molecule A and molecule B. We dissolve it in some solvent. We mix it together. We get a desired product, C. This is what we want to be making. And you almost invariably get a byproduct that you have to then dispose of. So in an example, so as we look at each of these reactants and each of these products, each one has its own specific hazards and costs. Both when we buy the reagents, we buy these things from wherever, some chemical vendor, and then we have to dispose of it. And as a rule of thumb, it usually costs between 2 and 5 times more to dispose of a chemical than it did to buy it. For obvious reasons. The regulations would be a major explanation for that. So here's a chemical reaction. So we take this molecule here, propylene. We react it with this molecule here called metachloroperoxybenzoic acid. In a solvent, we dissolve it in a solvent called methylene chloride. We convert this alkene to an epoxide here, and we get this byproduct. What I want to highlight here is that we start out with 6 atoms in this molecule and 16 in this. Here's the product we want. We went from 6 atoms to 7. We installed 1 atom. We are throwing away 15 atoms. Right? This is what we call a non-atom economical process. In other words, the molecules of the atoms in the reactants don't. Most of the atoms in the reactants end up mostly in waste. The rest, one atom ends up in the new molecule we're making. The second point I'm going to make is this is methylene chloride or dichloromethane. It's a hazardous air pollutant. It's extremely toxic. It's a great solvent, but it's from a toxicity standpoint, it's actually quite high. So we consider atom economy. In other words, how many atoms do we put in and how many do we desire to get out? And we have to think about how do we dispose of the stuff and how safe is it? So if I contrast it with this reaction, which is referred to colloquially as the Cliq reaction, I take a molecule such as this, which is 17 atoms, with a molecule like this with 14 atoms, with a molecule of copper in water. Reacted together, we get one molecule here. So these atoms all end up in the product, and we get copper back. Copper is a catalyst, so copper helps the reaction along, and then it can go back and participate on this side of the reaction again. This is a completely atom economical process. There is no waste generated. So copper is a catalyst, and water is sort of the granddaddy of green solvents, right? We know how to treat water. It's a safe solvent for workers, okay? So to take it to the next level, this is the process for making Zoloft. Okay, this is how this is made on the multi-ton scale by Pfizer. You do a reaction with this tetralone. You do what's called a reductive amination reaction. You do a reduction. You do a isomer separation. Then you do a deprotection, a salt formation, and then you isolate it. The chemistry is not as important except for these things. In red, I've circled the various aspects. The first is these reagents. So tin chloride it generates HCl hydrochloric acid when it contacts water. It's kind of toxic. Toluene and hexanes and THF, not exactly the safest solvents. THF as well generates what we would call peroxides. So sometimes on large scale, you can get very hazardous peroxides formed. And each of these steps is isolated, which means that they have to go through a process where they take the reaction and they do some process. They throw away the solvent. They get the new stuff that they have, they dissolve it in more solvent, and then do the next step of the reaction. They isolate it, they get rid of the solvent, they put in new solvent, okay? Very wasteful. The improved commercial route is much greener. You have the same, essentially the same reaction sequence, but instead of using these hydrocarbon solvents like toluene and hexanes, you're using ethanol. And this is not isolated. You carry it forward to the next reaction simply by adding palladium and hydrogen gas, which is a catalytic reaction, no waste generated. This next product is not isolated. You then do a isomer separation and then form the salt to get the final product. And so what I want to highlight is the solvents, our ethanol. Some of us had a little ethanol before we came in here, right? It's a green solvent. These are not isolated, so that you don't waste the solvent. You telescope the reactions, and eventually you do one isolation step. Okay? So as a result, this is the reduction in solvent use by the Zoloft process. So the discovery route used 232 liters per kilogram, and these are color-coded by nastiness. So methylene chloride, when the drug was originally discovered, methylene chloride made up just about a third of the amount of solvent used to make the material. If we get all the way to the end, we've actually only used 8 liters per kilogram, so we've gone from 232 to 8, and it's only using green solvents, ethyl acetate and ethanol. Okay? So this is the greening of the process. So think about the cost reduction in terms of the waste that's eliminated. You think about the reduction in toxicity and the reduction of risk. And so that's terrific. But that's great if you're a production chemist, right? So you want to make stuff on scale. You want to make kilograms of stuff. But if you're a research chemist where you're only working on tiny amounts relative to this, they're talking about thousands of liters of solvent. I use 10, right? So why, we talk about smaller scale, and actually people doing drug discovery on the early stage have a completely different mission that I'll talk about in a second. And so while we can't make huge differences in the amount of solvents we use, because we don't use that much, we have to make other kinds of changes to the way we practice our art. One way to do that is by what we call discretionary solvent usage. So chemists can decide, am I going to use methylene chloride for this reaction, or am I going to use acetone? Which is worse? Methylene chloride is clearly a worse solvent from a green standpoint. And so Pfizer implemented a solvent selection guide, which essentially had a list of bad solvents and a list of good alternatives. And just by communicating that to the discovery scientists, there was a 50% reduction in chlorinated solvent use across the entire research division, which at the time was enormous. And simply switching from hexane, which is toxic, to heptane, 1 carbon longer, not toxic, not as toxic, or switching from pentane to heptane. Pentane is extremely flammable, has a low flash point. So this change caused a significant reduction in worker hazard. Okay, so this is just discovery. So this is the small scale folks like me. And so what I'm going to go into now for the next hour of my talk is to talk about how another sort of approach for green science. How do we apply this to a discovery scale kind of environment? So first, a couple primers. The first primer is on neglected diseases. So neglected diseases are called that because they are generally diseases that affect the poorest parts of the world. There's no money for a drug company to recoup the $1 billion it takes to get a drug. There's no money available to recoup those costs. So publicly traded companies are not going to invest $1 billion in developing a product that they'll never recoup the costs for. And so as a result, these diseases are neglected. So what I have here is a list of various conditions, and this metric called the Disability Adjusted Life Year. What this is a World Health Organization metric for, it's a measure of quality years of life lost to disability or death due to one of these conditions. So for example, tuberculosis costs the world 34.7 million years lost to death or disability. Okay, a huge impact. In fact, TB, malaria, and HIV, which is not on here, represent the so-called top 3, the big 3. Other things, lung cancer, 11.2 million disability-adjusted life years. Prostate cancer, 1.6. And what I want to draw to your attention to is that if we look at prostate cancer, which is 1.6 million disability-adjusted life years, but we look at this disease here, African trypanosomiasis, or African sleeping sickness, you see it has roughly the same impact on death and disability. In other words, it's about 1.5 to 1.6. But if we look at the drug discovery pipeline, preclinical development, drug discovery, phase 1 and phase 2 and phase 3 clinical trials, you see prostate cancer has 34 programs as of 2008, 34 programs in preclinical development, 11 in safety phase 1 studies, 30 in phase 2 efficacy studies, and 3 in wider studies. If you compare African trypanosomiasis, there were 3 in preclinical development, 0 in phase 1, and 1 in phase 2. So here we have 2 diseases that affect roughly the same number of people in terms of death and disability. But because prostate cancer is a first world disease, that's where the money is, and that's where the money gets deposited in terms of research and development. Okay? It's not even a value judgment. This is just capitalism. This is the way it is, right? There are 3 diseases on this list that are caused by protozoan parasites, single-cell parasites: leishmaniasis, African trypanosomiasis, and Chagas disease. And these 3 parasites affect 22 million people per year, one way or another. They're not always fatal, but they're definitely disabling. Many times they're disfiguring. Of these, the World Health Organization has said that these protozoan parasites need to have new drugs, and I'll show you why in a second, including other diseases, including these worm diseases like schistosomiasis and bacterial diseases such as Buruli ulcer. African sleeping sickness is caused by a parasite referred to as Trypanosoma brucei. There are 2 strains. One causes the disease in the western side of Africa, and another causes the disease in the eastern side, so 2 different strains. You get this by a bite of a tsetse fly. So the bug comes and bites you. You feel kind of lousy for a couple weeks, and then it goes away. In sub-Saharan Africa, where you have to walk 20 miles to get to a clinic, no one goes to the clinic when they feel lousy. However, when the parasite is now gone from your bloodstream into your brain, you start to get this sleepiness and some behavioral disturbances, and that's when people go find out that they have sleeping sickness. It's 100% fatal unless you treat it. There are something like 50 to 70,000 people, according to some estimates, but that's a gross underestimate because a lot of people die from this without ever being diagnosed. The only way it can be diagnosed is by a spinal tap. Sub-Saharan Africa, field hospital, spinal tap. It's a bad, bad situation. So here we have this disease. Here are the drugs that people use. So the stage 1 disease, the ones that people seldom use because you never find a doctor at this stage, they work great, actually. These 2 are the ones you take for stage 2 disease, after it's moved into your central nervous system. Melarsoprol is an arsenic-based compound. That should ring some bells, even if you're not a chemist. It's an arsenic-based compound. We don't know how it works, and it kills 5% of patients. Okay? However, it works, and you don't die unless you're one of those 5%. And those who survive have side effects: reactive encephalopathy, heart arrhythmia, brain damage. Eflornithine is another drug which was developed originally for cancer. It's now used essentially for hair removal in women. Actually, it's a topical in women, for women to remove hair. It's a irreversible inhibitor. It was originally a cancer drug. It didn't work so well in cancer, but it clears parasites pretty nicely. But you have to take this drug for 3 weeks intravenously for 7 hours a day. So it's not like you just go, you get a shot, you leave. You lay on a bed for 7 days, and you get this drug infused into your veins. Melarsoprol, by the way, is corrosive. It has to be stored in glass because it eats through plastic. So imagine what an intravenous dose feels like. And that's like a 3 or 4 hour infusion. Grown men beg to die rather than have this drug. Okay. Clearly, in 2011, we can do better than this. So these neglected diseases are basically, the WHO has laid this out, and they say, look, it contributes to extreme poverty. It impedes gender equality. It's a huge cause of child mortality. In many cases, these are comorbid with HIV and AIDS and malaria. And frequently, people don't just have sleeping sickness. They have sleeping sickness and 3 other things. And as a result, you end up in this kind of death spiral of poor people getting poorer because they can't work. And then you just have this reduction in economic development, and it just keeps people poor. And so, from my perspective, the reason I decided to work in this area is because I feel like this is how we start to make a difference in relieving extreme poverty, is by doing drug discovery against these kind of diseases that we can do so well against first world diseases. That's the first primer, neglected diseases. If you don't know anything else after you leave today, realize how horrible these are and how much work needs to be done. The second point is, how do we do drug discovery? Typically, drug discovery goes like this. First, we identify a target. We find an enzyme or receptor that we find a link to a disease state. Enzyme X causes cancer, or a mutant form of enzyme X causes cancer. This is what we call target identification and target validation. We say this is a way that we can treat a disease. The second step is to find a compound that binds to and affects how that target operates. We test compounds by doing some kind of a biological assessment or an assay, and we test a collection of compounds. Frequently, we'll test on the order of millions of compounds to get a starting point. Then we do what's called compound optimization, where we make sequential alterations to the compound, or we make compound analogs to improve binding, efficiency, and many, many, many other properties that will get the compound so that it can be put into a pill so you can buy it and take it orally. Then it gets tested in clinical trial. Well, first it gets tested in animals to make sure that it works and it's not toxic. And then finally it gets tested in clinical trials. The timeline for this is roughly 6 to 8 years, and depending on the estimates you believe, it's on the order of $500 million to $1.2 billion to get a drug through this pipeline. Now, keep in mind that that number also includes the other 1,000 projects that failed along the way, right? We have to cover the cost of the so-called dry holes. But nonetheless, it takes a long time. So for a discovery scientist, your goal is to find a new molecule with utility as a drug. It has to be effective and safe and soluble. You have to have IP position. In other words, you have to be able to sell it. So you got to satisfy the lawyers. And you have to do this as fast as possible. You have to make stuff quickly, test it quickly, use that to inform your next round of designs, et cetera, et cetera. And so the goal for discovery scientists is to solve the major issues first: potency, selectivity, solubility, the very key aspects of drug discovery. And then you worry about the other stuff. The other stuff would be stuff like green chemistry. If I can't get a compound that's potent enough and can be taken orally, I don't really care how it's made, right? This is the attitude, and it's just a fact of life. And so a discovery chemist will cross out most of these things and say, I'll do the less hazardous stuff if it makes it faster. I'll design safer chemicals because, yes, I want safe drugs. I'll cancel all these guys out. I will do any of these things if it makes my discovery process faster. And inherently safer chemistry, I don't know. I'm going to use what works, and then we'll fix it later. And this is the general attitude, is that in discovery, green chemistry is really a process or scale-up chemistry problem. And so what I'm going to submit to you is that there's another way to look at this. One is to look at how we do medicinal chemistry and drug discovery. Like I said, choosing one solvent over another just by habit can make a huge difference in terms of your environmental footprint. Finding ways to purify compounds. Purification is the #1 source of solvent waste, is in purification. And if you find ways to purify compounds more efficiently, you reduce solvent waste. Using chemistry that works better, clearly. But from a strategic standpoint, we think about how do we make better compounds more intelligently. We use new assay technologies that generate less waste. And in the case that I'm about to show you, we use existing data by data mining and modeling. And we use a lot of the information that exists previously to drive the new project. So this leads into how we do neglected disease drug discovery. The first thing we do is we look for targets that are common between parasites that cause the disease and humans. So you have your parasite. The parasite's down here. This is the parasite. Okay, so we can now look at the DNA and we can sequence the DNA, right? We can identify, based on using parasite biologists do this frequently, they identify targets or enzymes that are important for parasites to survive. We then take that list of compounds and we compare it to enzymes that have been shown to be useful for targeting by drugs. A good example of this would be phosphodiesterase. Phosphodiesterase, as you might know, are the way Viagra works. We know a lot about phosphodiesterases, and it turns out that parasites have them too. Well, so we look at enzymes that are important for survival, compare them to enzymes that are important for drug discovery, you overlay them and you say which ones are in common, and then you reapply what we already know about the human targets to find new drugs for the parasite targets. Why do we do this? Well, we know that it's druggable. In other words, not all targets are actually amenable to having a drug target them. Not all enzymes can be inhibited by a drug that you can take orally. The second point is we can start with compounds without high-throughput screening. Frequently, programs start with a random screen of over 1 million compounds. Very expensive. If you have the resource, it's terrific. But we can do it with a few dozen selected compounds instead of a couple million. And we can reuse a lot of the data that was used in the human drug discovery program, such as what does the enzyme look like in terms of its shape and its binding site? What are the types of compounds that we know bind to this? What are the toxicity profiles? What kind of compounds can we make that are not toxic? And in addition, once we've identified compounds that are useful, we can then use that information to engage other people to help us develop the drugs. I'm never going to get a compound through the clinic. My development partners will. So I say, hey, Genzyme, we have some compounds that inhibit this enzyme for sleeping in trypanosomes that could be useful for sleeping sickness. Will you work with us to get it into the clinic? Frequently, most companies won't say, no. Most people want to say, oh, yes, we would like to work in neglected diseases. A lot of companies want to pick up the project after you've done all the risky discovery part. And then you're like, they go in for the glory, right? Which is fine. I don't really care. I'm not in it for the glory. So in sum, we're looking at it in terms of this being faster, cheaper, and greener. We don't run the big assays. We don't do a lot of the experiments that had to be done to repurpose it, so we should be able to be faster about it. And so when we compare the timeline that I just showed you, 6 to 8 years, we should be able to do it in maybe 4. How's that for ambition? The difference is, if we reduce the amount of staffing and reagent requirements, we test fewer compounds, so we have less assay waste. So instead of testing 1 million, we test 12. Not million, 12 compounds. Medicinal chemistry, you make fewer molecules, you generate less waste. And so this is sort of the operating model for our research. So in doing all these things, we reduce risk, reduce waste, reduce cost, and reduce time. This has been done. This is not our idea. HIV protease, this is a good proof of concept for this, what we call target repurposing. Excuse me. In 1985, so HIV was discovered in the early 80s. The genome of the virus was sequenced in 1985. 3 years later, they identified HIV protease as the important target. Now, smart people noticed that HIV protease had a similar structure to a drug target that had been well pursued for a long, long time, unsuccessfully, called renin, mainly for blood pressure. They have very similar structures. And as a result, they were able to repurpose the renin programs to get compounds into the clinic in 2 years. 2 years to go from validation of the protease target to getting compounds into humans. Okay? So a case in point, proof of concept. Now, there was a ton of money put into this, too. So there were a lot of people doing this at the same time. But nonetheless, it's proof of concept for us. And so I'm going to show you how we've done this now. In one particular case, we have a number of projects that use this approach. This one is the most advanced. This is an enzyme class, phosphoinositol 3 kinases, or PI3 kinases, and TOR, which is target of rapamycin. In humans, these enzymes control cell growth and metabolism. mTOR is important for controlling cell growth, cell shape, and autophagy, essentially cells deciding that they're done. And they're frequently extensively targeted for cancer and inflammation. And what this pathway is showing is, here's the pathway. So here are PI3 kinases. They feed downstream to AKT. They feed downstream to mTOR. There's a strong interplay in between them. And these gray boxes show drugs that have been brought to the clinic that target these. So for example, the compounds in this gray box here that target mTOR and PI3 kinases have gone to the clinic for cancer. Compounds here on the right just target mTOR. Compounds here just target AKT, et cetera. And so it turns out that Trypanosoma brucei, which causes sleeping sickness, Trypanosoma cruzi, which causes American sleeping sickness or Chagas disease, and Leishmania, which is another parasite that causes leishmaniasis, they have 12 proteins that have related enzymes. They include TOR. And so what these parasites look like, if you basically, this is a normal trypanosome for Trypanosoma brucei. When you essentially shut down TOR, in the parasite, you see it has a growth phenotype. It's much smaller. And if you knock down TOR2, it grows this so-called big eye phenotype, which is irreversible and they die soon thereafter. Leishmania has similar enzymes. We need the enzyme, the parasite needs these TOR1 and 2. TOR3 slows down growth, and you see these little white spots in the normal parasite. They get much, much smaller. These acetylcalcisomes are phosphate stores in the parasite. And so the idea is, if instead of knocking them down here like we're doing with RNAi or gene knockout experiments, we can knock them out with drugs, we should be able to get similar phenotypes, and then eventually the bug should die. And so we assessed known inhibitors, and there's a lot. There are a lot of known inhibitors in these PI3 kinases and TOR against cultures of parasites, and thereby validate these enzymes as a therapeutic approach forward, and then use this as a starting point for future drug discovery. There are some compounds that we essentially selected for this. We made a couple of these, but a lot of them you can actually purchase from third parties. These are almost entirely covered by other people's patents, be that as it may. So we take the compounds, and I sent it to 3 different collaborators. One collaborator lab tests against Trypanosoma brucei, and they are in Granada in Spain. The group at NYU, Ana Rodriguez's group, tests against Trypanosoma cruzi, and Steve Beverly's lab at Washington University of St. Louis tests against Leishmania. The idea would be we test them against the parasites, then we figure out how they work, and then we make new drugs. So it's a dense table. I want to hit 2 points. The first point is that we have compounds that show some activity against T. brucei, which causes sleeping sickness. These are pretty potent, actually, for a compound that we tested just right out of the box. Okay, these are they're probably about 10 to 100 times not potent. We need to get them about 10 or 100 times more potent. But if you look at this compound up here, NVP-BEZ235, this is a compound from Novartis. It's incredibly potent at killing Trypanosoma brucei, not just the animal infective strain, but also the human infective strain. It kills Trypanosoma cruzi, which I'll show you in a second, and it also is effective against Leishmania. This compound is a phase 2 clinical candidate for solid tumors. So Novartis has this in the clinic. It's already past phase 3, so it's safe. It's in phase 2 now, efficacy for solid tumors. And the question is, can it now be repurposed directly with no further work? This compound is out there in the clinic. Could it simply be repurposed for parasitic diseases like sleeping sickness? To just sort of drive this home, here's Trypanosoma cruzi. It lives inside human cells. So this, you can't see the outside of the cell, but this is the nucleus of the human cell. And these are the parasites. They're stained with a fluorescent antibody, and so you can see them just hanging out there in the cell. If you dose it with drug, they explode. Done. They lose their integrity entirely. This is possibly an interplay between the human enzymes, because again, remember, these compounds are really potent against the human enzymes, but they're also definitely hitting the parasite enzymes as well. We're not sure how they work, but boy, they work. This is the in vivo assessment. So this is in a rhodesiense, the human infective strain of T. brucei, dosed into mice. So you infect the mice, you dose with drugs after a few days, and then, so what we see here is the black is the control. So after day 3, animals are deceased. If we dose with drugs starting at this point, we start to see a dramatic decrease in the number of parasites that you can count in the bloodstream. It gets down below the detectable limit, and then you stop the drug dosing at day 6. Eventually, the parasites rebound. They come back, and they kill them. But the point is, we've extended survival by 4 to 7 days with 4 days of treatment. So 4 days of treatment with a phase 2 clinical compound. Okay? Big, big deal. And so we're talking to Novartis about this. They have mixed feelings. They're very excited about it because they have an interest in neglected diseases. But they're also nervous because if they find any problems with, so let's say they do a clinical trial in sleeping sickness, and it turns up some toxicity. Now their cancer indication is at risk, right? Because they have to tell the FDA about this. They have to say, uh-oh, when we tested this in people in sub-Saharan Africa, it actually had a little bit of a mortality problem. Suddenly, they've put their entire cancer portfolio there around this compound at risk. So they're understandably nervous, but they're simultaneously excited about it. So the point of all this is that, A, green chemistry is a different way for chemists to look at how we do chemistry. We decrease risks to workers, to the environment, and to the public, and we decrease costs. which doesn't seem like it should be true, but it makes a lot of sense when you think about the waste disposal and other costs that go with it. I hope I made it clear that while you can make huge impacts in manufacturing scale chemistry, discovery scale chemists can be more creative and put green principles into doing drug discovery. And then a green approach to drug discovery, target repurposing, to find anti-infective agents streamlines the process, reducing waste, reducing risk, reducing costs. And in my view, it's really directed towards sustainability because we're trying to improve the environmental and the economic situation of the poorest people in the world. And we did it by proof of concept in terms of these PI3 TOR inhibitors, and we've just submitted this work to be published. All the work, so these are all folks that I collaborate with all over the world. The folks who did this work are here at CSIC, Washington University, St. Louis, NYU, and the postdoc who worked on this in my lab is Caitlin Carver. Our work's funded by NIH, and we get a lot of free stuff from computational chemistry companies, and some drug companies do provide a lot of compounds and advice. So again, I thank you for the opportunity to speak. I'd be happy to answer questions either now or over coffee. Thanks.